Right. Hopefully it's in presenter mode for all of you now. So as Heidi and Sharon mentioned, my name is Jules Savat. I'm a genetic counselor at Geisinger, where I help to coordinate ClinGen's patient data sharing efforts, which includes Genome Connect, our patient registry, as well as some work with other gene and disease-specific registries. So I'm really delighted to speak to you about the ways that we're engaging patients in data sharing. So today, to give you a sense of what I'll talk about, I first will talk a little bit about how patients can participate in the data sharing process by submitting their genetic and health information to patient registries, including Genome Connect. We'll discuss how patient-shared data can benefit both the genomics community as well as individual participants. And finally, I'll end by outlining how you can get involved by either using patient-provided information or encouraging such data sharing. So similar to Heidi and Sharon, I think I'll take about 20 minutes for my talk, so we should have plenty of time for questions. So feel free to drop those in the chat as you think of them as I'm talking, and then we'll address those at the end here. Before I go ahead and launch into my slides and telling you about Genome Connect and patient data sharing, I thought it might be helpful for me to know just a little bit about who's all here and your involvement with ClinGen. So I'm going to go ahead and drop a link into the chat. But you also can use the QR code to answer that the link. Let me put this here. So the link is in the chat. Let me put it back on the screen. And I'll move over. Thanks for the audience participation. I know it's late in the day on Thursday after listening to lots of talks. Give it just a couple of moments. Well, it looks like, I'll let these keep coming in, but we have sort of a diverse group here. Some involved in VSEPs, others GSEPs, others just here to listen and learn. So hopefully there'll be some takeaways from today. And I saw there's not an option for I don't know. That's okay. If you don't know, I think we'll be able to address pieces of how patient data sharing can potentially impact your work that you're doing or that you might do in the future. So you've seen this graphic quite a few times today already, but I think it's useful just to explain what we're going to talk about today, how it fits into the broader ClinGen effort. So I think Heidi and Sharon talked a lot about ClinGen's critical questions, and Heidi alluded to the fact that those efforts really rely on crowdsourcing, on data sharing. She talked about how ClinGen is working with ClinVar to encourage data sharing from labs, clinicians, and researchers. And as she mentioned briefly as well, we also encourage patients to share data. And that's really what we're focusing in on here. Labs, researchers, and clinicians have been sharing genetic and health information for some time, and Heidi put some numbers around how many variants have been shared. But ClinGen felt strongly that patients could also serve as an important source of additional information. Patients are often experts about their own health history. They can provide much more details about their symptoms over time, their diagnoses, and their health. And ClinGen felt that access to that additional information could be helpful to increase our understanding of genes and genetic variants. As a result, back in 2014, the ClinGen team launched a patient registry called Genome Connect, really to empower patients to share their de-identified genetic and health information with publicly available databases like ClinVar. Genome Connect is open to anyone who's had genetic testing regardless of results or diagnosis, so it's not gene or condition specific. Participants enroll online, consent to de-identified data sharing and recontact from the registry. They provide their health history through patient surveys, and they upload a copy of their genetic test results to their online account. As I mentioned, we collect health history via surveys. So after consent, every participant receives an initial broad health survey. This survey inquires about general issues across 17 sort of broad categories or body systems. So things like issues with the cardiovascular system, issues with the endocrine system, GI issues. Each question provides examples of issues in that system. And then for those that individuals indicate they've had an issue with in the past or currently have an issue with, they then are assigned additional questions, trying to drill down further what are their particular symptoms and diagnoses. We also assign additional surveys over time to collect even more information about their health history. So for example, an individual on one of the first pages of the survey would see a question about, have you had issues with your heart and or blood vessels? A simple yes, no, unsure. If they say yes, they receive an additional page that lists out cardiovascular issues, arrhythmias, heart defects, cardiomyopathy, which you see here on the screen. Each individual phenotype is written out in patient-friendly language, but on the back end, if they indicate, yes, I have a history of cardiomyopathy, each of these symptoms is mapped to a human phenotype or HPO ontology term, which allows structured data sharing outside of Genome Connect. In addition to this initial survey, individuals who say, yes, I have a cardiomyopathy, would then receive a second survey later on, asking more questions about the type of cardiomyopathy, any additional risk factors that are non-genetic, like a history of chemotherapy, again, to try and collect more phenotype information, more information about their health. Participants share their genetic test results by uploading either a PDF or image of their result to their account, or by providing our team with permission to request their records from the lab that performed their testing or their healthcare provider that ordered it. Genetic counselors on our team then review those reports to ensure that all of the genomic data are collected in a structured and standardized way, and really to ensure its accuracy so that we can then go share that information more broadly. Once genomic and health information are collected from patients, their de-identified variant and health data are shared with ClinVar, which Heidi mentioned, and you'll hear much more about from Melissa shortly. But as you probably know, it's a publicly available database of variants and their classifications. And as you also know, this is where labs, clinicians, and as Heidi reviewed, often submit variants. So submitting information here from a patient registry makes it publicly accessible to those other stakeholders so that they're aware that this variant has been identified and potentially can see more detailed health information about the individual that was found to have that variant. The data are submitted to ClinVar using a phenotyping only submission type, which highlights that this information is coming from someone who's not reinterpreting the variant, didn't identify the variant through clinical testing, but is submitting it as it was reported by the clinical laboratory, but along with additional phenotype information. This submission type isn't counted towards star status or aggregate classification, which prevents a duplicate counting of a single patient. And I'll show you later on some slides, but we do all we can to try to highlight reporting institution's classification, so a ClinVar user can readily see this lab submitted, and also that participant must have gone and enrolled in Genome Connect. In addition to submitting to ClinVar, we submit some genes to Matchmaker Exchange. A subset of participants share reports that identify candidate genes, and as you might know, candidate genes are those that may be associated with a set of symptoms based on some preliminary research, but really much more information is needed to better understand if that particular gene is associated with a Mendelian disease. Candidate genes that we identify on participant reports are submitted to Matchmaker Exchange, which is a data network that's really working to enable matching with other healthcare providers and labs who are exploring gene-disease relationships. Our hope is that by sharing information here, we can contribute to moving these candidate genes into a more definitive category of having a disease association or not being associated with Mendelian disease. So why is patient data sharing important? So I certainly have many a slide to talk through our experience to date, but I thought it might be helpful at this point, since I'm halfway through, for some other participation from you all. So that same link that's in the chat, this is a word cloud. You can go ahead and type in if there are any initial thoughts you have about how could this information be useful. And if not, don't worry, I'm going to tell you in the next few slides here. Give it another second. There we go. Some are coming in. Support, connection, improved precision medicine, genome studies, genetic mastery, discovery. So I think you've hit it with these, you hit the nail on the head, that benefits are sort of twofold. And as I alluded to, I think earlier in our discussion, that patient data sharing can be useful to the genomics community, lead to discovery. Also, there are some benefits back to individual participants like connections and support. So I want to talk through those a little bit further in these next few slides. So first, how can patient-provided data inform our genomic understanding, our understanding of variants and gene disease classifications? So I think Heidi summed this up well, but when a genetic variant is identified, the critical questions really are first, is that gene associated with disease? That's our gene curation efforts are focused on that. And then second, if so, is that variant pathogenic? Is it going to impact that gene's function in the body? Patients participating in Genome Connect, what we've seen over the past nearly 7 years is first that they're providing additional phenotypic information. This was really the goal of Genome Connect from the outset, that patients could provide much more details than are often included in lab or other submissions. We're seeing that in practice. They're providing health history data, they're providing information regarding inheritance of the variant, and we also have the ability to recontact them over time if additional information is needed or could be helpful to inform our understanding. Really knowing patient-specific symptoms, knowing the inheritance of a variant, knowing if other variants were identified through testing, all of that can help shift variants of uncertain significance into more definitive categories or could inform a gene-disease relationship, perhaps. In addition to providing additional details like enhanced health information and information regarding inheritance, patients enrolling in Genome Connect are also providing novel genetic changes that haven't been submitted to ClinVar previously. From our submissions to date, 41% of variants weren't submitted by the reporting laboratory or another institution previously. So really that participant taking the time to enroll in the registry is what got it out there into ClinVar. Putting this information out there in the public space can be useful the next time that particular variant is identified or if a similar variant is identified in future patients. Altogether, patient data sharing can provide additional data that can inform both genes and variants. So if we're thinking about those gene curation and variant curation efforts, patient data sharing can provide access to unpublished cases that could be used to inform gene disease relationships. Patients also can provide case level details that can be used to inform variant classifications. To help illustrate the power of patient data sharing, I wanted to share a brief case example that is actually something that occurred and that we can appreciate from our data sharing. So for the purpose of this case example, I will call this participant Ella. She's a pediatric patient with a history of seizures beginning in infancy, and she had exome sequencing due to her history back in 2015. A likely pathogenic variant in SCN8A, a gene associated with the seizure condition, was found. Her parents were tested as part of her trio exome, and the variant was found to be de novo. Because the variant had not been seen in healthy individuals, was found to be de novo, was expected to impact the protein, the clinical lab classified it as likely pathogenic. Ella and her family had an answer for her history, but they chose to participate in Genome Connect to share her information more broadly. We then submitted her de-identified genetic and health information to ClinVar. At the time of our submission, there were 2 other records in the database. So lab 1, the lab that had actually reported the variant out to this family, had also submitted to ClinVar, though their record contained fewer details about her clinical history and didn't have information about inheritance of the variant in her family. Labs often have these details, but they might not be able to share them with ClinVar. We also saw a submission from Lab 2, who had classified the variant a year prior as a variant of uncertain significance. After our submission, Lab 2 reached out to the registry team after seeing our ClinVar record and wanted to know more about Ella's seizure history and to confirm that the variant was in fact de novo. In their patient, parental samples weren't available, so they weren't able to complete parental testing. And I think, as you all know, understanding if a variant is de novo can be incredibly helpful in informing that variant classification. So we were able to confirm by re-reviewing Ella's report that the variant was in fact de novo, which prompted Lab 2 to reclassify the variant as likely pathogenic and update the family that they had identified that variant in. Ella's results in care weren't impacted by her family taking the time to share her results, but ultimately them sharing enabled another family to get clear genetic results. So in addition to informing variant classification and gene disease validity, patient-centered data sharing can also benefit the individual patients themselves. So first, it can facilitate genetic updates. So as I just outlined in that case example, our understanding of variants can shift over time. In fact, we hope to reclassify variants of uncertain significance, and rarely we also know that our understanding of pathogenic and benign variants might shift as well. In just one study of a cancer genetics lab, over a 10-year period, 6.4% of variants were reclassified, leading to more than 60,000 updated reports. So we know this happens in practice. Clinical lab and healthcare providers' duty to reassess genomic results and recontact patients has been the subject of ongoing discussion, and there's no clear legal duty to recontact, but there is a consensus that recontact is beneficial, and most patients seem to value being recontacted, hearing about updates to their results. Lab and healthcare providers' reassessment and recontacting practices really vary, and there are certainly barriers to recontact, including limited resources, limited reimbursement, changing contact information of patients. So under current circumstances, two large genetic societies, both ACMG and ESHG, have suggested that patients share in the responsibility of recontact. ACMG has also suggested that novel methods of recontact are needed to ensure that patients are remaining informed of their results. So early on in Genome Connect's data sharing, we recognized that the registry might identify cases where the participant's report is out of date compared to the current knowledge base. Whether that's in ClinVar, we can see that the reporting lab has updated their classification, or via our submissions to Matchmaker Exchange, we might come to appreciate that a candidate gene is now disease-causing. As a result, we reached out to our participants with a survey back in 2016 to just learn a little bit about would they want to receive updates from the registry, what are their thoughts and reactions to that. After an overwhelmingly positive response that participants wanted to receive updates in general and wanted to receive information from Genome Connect, we designed a process really based on their feedback. That in February of 2017, we prompted all prior participants to provide their preference regarding the receipt of updates. And now upon enrollment, anyone who's newly enrolled is required to tell us if they'd like to receive an update from us, if we should identify one from our data sharing. Of course, we won't identify all updates, and that's all in our consent language, but we'd like to give the choice that if they would like to receive this information, that we will pass it along should we learn of anything. To date, 98% of those who responded and said, provided a preference have opted to receive updates from the registry. Should we identify an update to results, we first reach out to the reporting laboratory if needed to confirm the variant classification in ClinVar or that gene disease relationship. And then from there, reach out to the participant, informing them that there might be an update to their result and encouraging them to reach out to their physician or genetic counselor or other provider to learn more about that particular update and its impact on their care or their results. In total, Genome Connect has provided 45 updates back to participants to date. They're outlined here on this slide. 17 such updates that fall into that medically significant or potentially medically significant bucket that Heidi had alluded to. So it might impact diagnosis and or management. So ultimately, patient data sharing can enable individuals and families to remain informed about their genetic test results. Finally, patient data sharing can support connections between stakeholders. So I think as some of you in that word cloud thought of, registries can connect others. So first, Genome Connect enables connections with researchers or clinicians who are interested in a particular gene or variant. If a researcher or clinician reaches out to us, we're able to recontact participants to collect additional information or to inform them of additional research opportunities. And additionally, participants can connect with one another based on gene, disease, or U.S. state through the registry. Many genetic conditions are rare, and patients might have the experience of being told that they're the first patient their healthcare provider has seen with that diagnosis or a variant in that gene. And the registry can really enable them to connect with others to exchange both informational and emotional support. Given Genome Connect's experience with patient data sharing, I think we really recognize that patient registries could serve as an important source of additional information while also benefiting the individual participants. We also recognized that Genome Connect is just one of many patient registries. There are other gene and condition-specific registries out there that are potentially collecting really valuable information but might not be sharing it in a public way. As a result, in 2018, we started working with registries who were engaging patients with genetic testing or genetic diagnoses to really use the process that we developed to share data with ClinVar and help them do the same within their registry. We work with registries that are on the same registry platform as Genome Connect through Invitae, as well as registries on other platforms. And to date, we've engaged with 10 registries to help them submit to ClinVar as well. Altogether, Genome Connect and this work with other registries have allowed us to engage nearly 5,000 patients in data sharing, and more than 1,900 variants have been submitted to ClinVar, and I will say we have a large submission underway, so we expect that number to tick up soon. So finally, how can you get involved? And regardless of how you're involved in ClinGen. So first, you can use the information shared by patients to inform the understanding of the relationship between genes, variants, and health. The variant and health details that we collect from participants are periodically shared with ClinVar, and I'll walk through a ClinVar record here briefly just to show you what it might look like. So use that if you're involved in ClinGen efforts or if you're involved in caring for patients. That's something you can use. We're also exploring a process to export data for relevant VSEPs and GSEPs. So if you're involved in a VSEP or GSEP, we might have been in touch with you already to highlight potentially genes that have been curated as having limited, refuted, or disputed evidence that we have participant cases around. Or if there's a VSEP where we have participants with variants in that gene, we might have been in touch with you already or told you a little bit about what we have. So just briefly, a ClinVar record here. So if you search a variant and come to the variant page, if you scroll down, you might see a record from Genome Connect. So as a phenotyping only submission, we're not interpreting the variant ourselves, so we do not provide a classification, but we do provide information about how the reporting laboratory has classified that variant. On the main page here, that information can be found in the comment. So you can see that it was interpreted as likely pathogenic the day it was reported, and then the GTR ID, the institution that reported this out. Inheritance information, if we have it, also can be found on this main page. And then much more detail can be found if you go to the supporting information. And then if you choose your columns, you can see all the information that we've submitted from participants, including their HPO terms and age range, sex, so all on this page. We also welcome questions, so it's not uncommon for us to get questions from those who see a variant record, and we're happy to answer that information. To find all of the submissions from Genome Connect in the registries that we work with, you can go to the submitter list on the ClinVar website, and anyone that we've helped facilitate a ClinVar submission from will have Genome Connect in the name. So our submission is Genome Connect. If it's another registry, it'll have their name and then ours as well. Additionally, you can work with us to expand patient data sharing. So if you work with patients, inform them of opportunities to share data through Genome Connect. We have a number of resources on the ClinGen website to help you do so, and many clinical laboratories include wording about the registry directly on reports. If you work with a patient registry, discuss the opportunity to engage with us in what we call our patient data sharing program, so how we help them submit data to ClinVar. So in summary, I hope today in my time that you all have come to understand that Genome Connect and other patient registries offer a structured way for patients to share data. And that information can be useful because it provides additional details as well as novel genomic information that can inform our gene and variant curation efforts. Also, registries can serve as a liaison between labs, researchers, and patients to ensure that patients are remaining informed of their genetic test results, and also that we can pass on information about additional research opportunities. Finally, ClinGen is actively working to continue to expand patient data sharing efforts by working with more patients and other registry groups. So with that, I saw the chat was active while I was talking, so I will acknowledge those who make this work possible, including participants and the team at Geisinger as well as the Broad. Our general email is up here on the slide, and why don't I stop sharing so I can look at the chat and hopefully answer the questions that you guys might have.
The good news is that Melissa was active in answering some of those questions, one of which relates to her own talk. So and one was just defining a de novo variant.